I'm happy to report some of our latest work on trying to stop a big-headed carp migration up the Mississippi River. And so in conjunction with my graduate student, Michael Frett, we've been evaluating the effects of carbon dioxide and sound in a model lock and dam. And so we know both sound and carbon dioxide are fairly effective deterrents against big-head carp migration. However, they both have their positives and negatives to them. As far as CO2, we prefer not to pump CO2 into the environment consistently. The other way, also sound may, after some time, the fish may become habituated to it. And so we decided to combine the two using classical conditioning. And here we test the fish with CO2 and sound, and then later on just with sound. And the thought is that by classically conditioning the fish, they will associate the onset of sound with CO2, which they really don't like, and they will leave the area. And so we believe this strategy will increase the effectiveness of both deterrents and also decrease the need for using carbon dioxide on a daily basis. And so our targets are locks that are adjacent to dams in the Mississippi. The dams are fairly decent barriers to upstream carp migration. However, the big-headed carp can pass through the lock chambers and head upstream. And so this is the theoretical framework of what we were doing. We originally had just proposed using sound in the lock chambers, but we believe by the addition of CO2 will make it a much more effective deterrent. And so in this case, what you're seeing is the downstream lock opening, and we're adding CO2 with the sound, and we would run this for several days. And then during subsequent tests, we would just use the sound only. And we think this will be extremely effective. And of course, we may have to reinforce this occasionally with CO2 addition. This is the overhead view of our model lock and dam. We have an upstream pool and a downstream pool that is separated by a dam. And the only way the carp can get from the downstream pool to the upstream pool is via a lock chamber, which has an upstream gate and a downstream gate. We've embedded 2 underwater speakers into the lock wall, and we also have the mechanisms of bubbling CO2 into the lock chamber itself. These are just illustrations of the lock and dam. Our colleagues at Salpal were very kind enough to construct this for us. In both these cases, we're looking from downstream to upstream. So in the foreground is a downstream pool, in the background is the upstream pool, and you see the lock chamber on the left. And to give it some scale, those are our 2 undergraduates that worked with us all summer standing behind the tank. And so we've learned with previous experiments that it's very important to concentrate the sound and make it be a very sharp gradient. So we really worked hard to restrict the sound just to the lock chamber, and we were quite successful. So the sound does not bleed across the walls or very far downstream. We're focusing on 2 to 4 kilohertz sound. This sound is very effective at deterring carp. However, it does not affect the hearing of native game fish, which mostly cannot hear above 1,000 hertz. There are some native fish that it will affect, but by using these higher frequencies, we are not impacting all native Minnesota fishes. So this is the experimental setup or schedule. We have schools of 10 bighead or silver carp. We acclimate them for a couple days with both gates open. Then we do 6 conditioning trials over 2 days in which they're placed in the lock chamber with both gates shut and presented with sound and carbon dioxide until equilibrium is lost. Then we start the experimental trials, and those take 7 days. We operate the lock just as it is done on the Mississippi in the same procedure 4 times daily. When the downstream gate is open, we play sound for 30 minutes. We then close the downstream gate, open the upstream gate, and then the upstream gate closes after another 30 minutes. This video with the very annoying sound playing in the background, that's the sound we use, just illustrates how this works. For the most part, the carp school in schools of 5 to 10. Here, there 8 have decided to try to go upstream, and we have 2 outliers. And just so you remember, the sound really does not start until just at the beginning of this lock chamber. So right now, they're probably not detecting the sound, but the school is going to make a move upstream, probably to get to their preferred upstream pool. And as you see, as they just about starting to hit the sound gradient, they will retreat very quickly and go away from the sound and do not enter the chamber at all. And here they are coming out because they did not want to go into the chamber. We have at least 2 cameras operating, and this one is just a close-up of the lock and dam, so we can see what's happening on a smaller scale. Again, the sound is probably starting around the gate area, and you'll see a few fish poke their noses up into this area and then turn around. So here they come. And they have no desire to go past the lock gates into the lock chamber. And so here's our preliminary data. During control periods, when both gates open, the carp just love heading upstream, and they spend the majority of their time in the upstream pool, little less time or much less time in the downstream pool, but they're not intimidated by going into and through the lock. And sometimes they actually stay sometimes in the lock. So the lock is not a barrier to them at all. They don't rush through it. They just slowly swim through the lock when they're moving from one pool to the other. And this is our first data. This is with the lock in operational mode. And as you can, so this is 7 days, 28 trials, and the fish do not want to go in the lock at all. They avoid it very well. There's occasional outlier that may stick their nose in the lock, but for the majority of the time, even though they do go towards the lock, they do not enter the lock. And so these are fairly exciting results, but I must caution you that they're right now preliminary. We've only done 3 silvers and 1 big head trials, so that's N of 10 for each of those trials. But it's clear that in these 4 trials, the fish avoid the lock chamber for 7 days after conditioning, and we've had 0 upstream transits. And so we're going to continue this work for statistical purposes. We need to finish up these studies. So we need 5 trials for the silvers and 5 for the big head, but we're well on our way, and we should be finished that by the end of the fall. And then we want to look at the effects of this deterrent with motivated fish. And the way to motivate these fish is by food. And so we'll be putting food in the upstream chamber only and seeing if the fish are motivated to go through the deterrent to get the food. Also, we know that the locks operate almost year-round, and at least in the ice-free months. And there's certainly changes in temperature, and that does affect carp swimming ability and behavior. So we would also like to do trials at higher and lower temperatures. These trials were done at 20 degrees. We'd like to go at 15 and 25 degrees. And so Mike has been instrumental in doing all these experiments. I'm from, we're both from Minnesota Duluth. However, he's now resident in the Twin Cities, and he's a water resources student that's doing all the study. Jessica Kazarik has been our very good colleague from Southall, is helping with some of the flow measurements. And we've had these two great undergraduates work for us during the summer. And of course, none of this could be done without the support of Southall and MAISRC staff. So thank you for listening, and I'm happy to answer any of your questions.
Thanks so much, Al. One of our first questions says, will this deterrent need to operate year-round?
I don't think the deterrent will need to operate year-round for several reasons. One, the locks do not operate during the ice, when there's ice on the river. And two, I have a feeling that there will be much less movement in the cold weather, when the water gets cold, than in the warm weather months. So I don't foresee it operating year-round. However, if this is successful and some of the southern states want to use it, it certainly has the ability to operate year-round.
We have another question here that says, do you anticipate that the conditioning would be effective only with those individuals that have been exposed to both sound and CO2? New individuals or offspring might swim through?
That's a great question, and we will try to address that once we finish our future plans. But what we've seen is these are very herd-oriented fish, and if the majority of the fish are moving away, we do see that fish that have not been conditioned yet will move along with them. So yes, it's a concern. It's a very good question. But if we could just postulate how frequently this may need to be done, maybe the carbon dioxide needs to be reapplied every 2 weeks, and so hopefully those new arrivals in the area the sound alone will be effective, and then they'll come to associate sound with carbon dioxide and make the subsequent sound projections even more effective.
And we have one more question here that says, will you test some native species just to confirm that they are not affected, or do you already have that data?
We have some data done at USGS where the native species were not affected by this sound, or at least we couldn't see any movement correlated with the sound. However, we have not tested that in the lock and dam, and we certainly would like to. Unfortunately, we only have one model lock and dam. As you can see, it's a very large tank, and it takes Mike about 2 weeks to run one trial. And so our ability to run multiple trials is not possible at this time. So we are concentrating on the silvers and the bigheads, but it may not be out of the question to try to kill 2 birds with 1 stone, and maybe put natives in with the silvers and see how they react. So that's something we could certainly look into doing in the near future.
And we have time for just one final question, which is, based on your preliminary results, do you have a recommendation for lock operation on the Mississippi River?
Yes, that's a very good question. I certainly would try to keep the downstream gates closed as long as possible. And I understand that this is a shipping issue and not a biology issue, but every time those downstream gates are open, they allow carp to potentially go into the chamber. And we have been at the locks, and they open well in advance of the ships. And so and I certainly don't want to violate any safety protocols. But if the downstream gates could remain closed for longer periods of time, as long as that's not endangering shipping or individuals, that would be certainly our preliminary recommendations just based on this study.
Thank you so much for presenting. We have no further questions, and we can move on to our next lightning talk. Thank you so much, Al.
Thank you.
Thank you all for coming to listen to my talk. As Ethan said, I'll be talking about the effective rusty crayfish removals to protect wild rice, and I'll be talking about year 1 of 2. I need to mention that Josh Dumke has been the principal investigator for this project since the beginning, and he went through the entire field season, but he has since taken another job elsewhere, so this project is up for someone else to manage. And I was someone that slipped on a rock in the lake and fell, so I flooded my waders, and that is what gave me the credentials to give this talk. I work for Soil and Water in Grand Marais, Minnesota. Here we know that rusty crayfish are invasive. They are native to the Ohio River. So when they came to Minnesota, they outcompete our native crayfish for resources and shelter and habitat. And wild rice is in most of the lakes that rusty crayfish are invaded into. There's about 46% of all the lakes that rusty crayfish are in are also wild rice lakes. And we had 3 different lakes that we sampled this year in St. Louis, Lake, and Cook County. 2 objectives for this project were to determine the most effective trap and bait types for rusty crayfish removal, and then to measure rusty crayfish migration rates using a mark and recapture method. So trapping was conducted twice per lake. So 3 lakes were sampled in the spring, and then 3 lakes were sampled in the summer, in August specifically. And there were rusty crayfish that were marked and then released in the springtime, but all of the crayfish that were trapped in August were removed from the lakes. And this picture shows Brennan with a lot of the traps that we used. The trap types that were used are the standard minnow traps, and then an extra large minnow trap. So it just has a middle piece that allows it to hold more crayfish in it. And then the C option is a refuge trap that doesn't have any bait in it. It just simulates habitat and safety shelter for like the PVC tubes for the crayfish to just hang out in. And then we also use these pods, which had 3 different baited traps together sitting in one little group. The bait types were the delicious meow mix in the seafood select flavor, giving a little chef kiss to it. And these came in a plastic cup with a tinfoil lid on them, like a yogurt tab. So for these, we slit 2 or 3 holes, like slices into the lid, and placed them into the trap. So the crayfish couldn't really get inside and eat the cat food. It was just like seeping out little smells. And then we have smelt from a VHS free certified bait dealer. And then some Oscar Mayer wieners were also used as the bait. And so for the smelt and the hot dogs, they also had a bait pill in the traps, a little picture down here. So we put an entire fish, an entire hot dog in the traps that were loose, and then a chunk of fish and a chunk of hot dog separately were placed into the bait pill, so that once the crayfish ate all of the smelt or hot dog, there was still some bait in that little container that they could smell and try to get food from. And that the idea was that would keep the crayfish in the trap for longer instead of them trying to find their way out. Second objective is about the rusty crayfish movement. So each of the 3 lakes had an area that was the removal zone, and that is where the vegetation, the wild rice was. And then they also had these mark and release areas that were designated for where the pods would be and where the crayfish would be marked and then released. So this bottom picture on the left shows the orange dots as the removal site for all the crayfish. And then the blue dots are the pod sites, and that's where crayfish were marked and released. So the middle picture shows how we were marking them, just a little pin in their tail fins. This top right picture shows there's a mark in the telson, the middle fin, and then there's one in the left middle uropod. So that using this chart that we followed, that means that that crayfish was marked on the left shoreline at pod number 2. So going back to this map, it would be this pod is where it was marked. It's just a map of the 3 different lakes and the pod locations. Some pictures of out in the field. We had to wade to a lot of the sites because they were shallow. This bright picture shows pulling out a rusty crayfish trap, all the crayfish inside, and we dumped them onto the sorting tray. And this sorting tray has these bars that are about 5/8 of an inch apart. So any of the crayfish that were smaller than that width, they would fall through into the tub. And those ones were considered small and they didn't get marked. We just sexed them. And then the larger crayfish that stayed on top, if we were at a pod location for marking, then we would mark those larger crayfish and release them back into the lake in the spring. The left picture has a crayfish in the refuge trap hiding in there. Middle one is another sorting tray with the bait pellet. And here on the right shows 71 pounds of crayfish that was removed from the lake on one day. Brennan trying to lift it up and weigh it. Here's an action shot of the trap underwater. You can see they're the underside of one of these crayfish with their giant claws, and then the hangout of the other crayfish trying to figure out if it's worth it to get inside there. Now here are some numbers from our work. These 2 graphs show the number of crayfish caught per trap type. The one on the left shows by sex, male yellow, female green, and then the graph on the right shows by crayfish size, small blue, large orange, and the bottom shows you how to identify the sex of the crayfish, female with eggs, female, and male. So each of these crayfish that were caught, we had to sex them. And the overall takeaways from this slide shows that refuge traps are not ideal for removing a lot of rusty crayfish from a lake. And it also shows that the extra large traps aren't really necessary. We don't need that extra middle piece. The standard traps work perfect. Here's a graph showing the total number of crayfish caught by the trap type and arrangement. So this one adds the pod into it. So the takeaway from this shows that standard traps work really well when they're baited, and the pods work even better. So if you're going to try to remove a lot of rusty crayfish from a lake, do it with a bunch of traps in one location to try to attract all of them. This graph shows the total crayfish caught per lake by the bait type. So you'll see that the orange bars are from Caribou Bigsby Lake, and there was a lot more crayfish caught in that lake than the other 2 lakes. And that can be for several reasons, but I think one of the main ones is that Garden was the first lake that was surveyed in May, and then the week after that was Dumbbell, and then Caribou was next. So Caribou had the advantage of knowing what not to do from some mistakes that could have been made on Garden and Dumbbell. So we were able to catch a lot more crayfish by not doing some things like trapping in rock sediment instead of in silt sediment. And then just looking at these bars, it shows like cat food is not really the ideal food source for crayfish, but they like hot dogs and they like smelt. But overall, this doesn't show us a lot of obvious preference. We need more data to show us another year of experience to really make any decisions. So here is what worked for this year and improvements for next year. And this is all coming from me, not from the crew as a whole. So I think that the bait pills worked really well in the traps. They kept the crayfish in the traps when the rest of the bait had been consumed. And using GPS points for each trap location obviously was a great choice, but then also clearing the boat's track each day so that you can tell which traps you went to already and which ones you had missed. Using clicker counters for counting the crayfish was essential because at the caribou, there were hundreds of crayfish in a trap. So there was one person keeping track of how many males and how many females, and then another person that was just picking up the crayfish to sex them. So to have the clicker to say male, 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 female, male, 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 that was needed. And having radios so that we could talk to each other in our separate boats without having to drive over. Some improvements for next year could be having better organization of all the equipment in the boat and in the storage. There's just a lot of traps and very small boats to do some of the work in. And one thing could be swapping the extra large traps for the standard traps, since the extra large weren't really necessary. The standard traps worked just fine. And then for the refuge traps, we this year had checked them every day, and perhaps they could be set out for the entire week for 3 nights, and then checked at the end to see if that allowed more crayfish to enter, if they felt more safe in it, if it wasn't moving every day. And also we need to decide if hybrid crayfish are considered native or invasive for this project. Because hybrid crayfish was not discussed at all before we did the field work. And we found some hybrids in Garden Lake. So we considered those to be native since they weren't discussed previously. And here shows the difference between rusty and the hybrid. On the left is rusty crayfish with the telltale rusty patch on the carapace. And then on the right shows a northern clearwater and rusty crayfish hybrid. This crayfish still has the black bands on the tips, just like the rusty does, but it doesn't have a rusty spot on the carapace. And northern clearwater has a dark line on the tail, dark band, and that's present on this hybrid. So it's obviously not a rusty, and it's also not a clearwater. Oh, it's a hybrid. So the first year impressions for me is that bait preferences are not obvious. And our spring sampling, the water temperature was very cold. I mean, ice out was 2 or 3 weeks before we did our first trapping. So the crayfish were very slow moving and not very hungry due to mating and molting. And August trappings were the most effective for harvest and removal. So if you're a lake that's trying to remove as much rusty crayfish as possible, August is the time to do it. And the crayfish migrate a lot farther than anticipated. There was one crayfish in caribou that was observed. It was trapped in one pod, and then it moved 250 meters overnight and was trapped in another pod the next night. And I need to thank everybody that helped with this project so far, and all of the funders, all the organizations that helped fund and staff some time for this project.
Thank you so much, Amanda. And unfortunately, we just have time for one question. And the question is, did you make any direct observation on rusty crayfish impacts to wild rice?
We didn't get that far in the project yet because we weren't able to trap in the wild rice areas because the sediment in the wild rice areas are full of silt. And when you trap in silt, the traps just sink to the bottom, so crayfish can't enter them. So that is something that we're trying to work on for next year, but knowing that how to effectively trap them in areas near wild rice helps to know how to remove and lower the population.
Awesome. Thanks, Ethan, and thanks everyone for tuning in. As Ethan said, my name is Abha. I'm presenting today about our upcoming 2024 MAISRC project on cultivating resilience, native revegetation for AIS resistance. This is our team listed here. This project's led by Dr. Dan Larkin, along with Naomi Blinick and Dr. Mike Verhoeven. So the impetus for this project is related to the reinvasion of invasive aquatic plants and how they are related to native plants in lakes. So a lot of times, hundreds of lakes across Minnesota are herbicide treated for invasive aquatic plants. And in an ideal world, we see a bunch of invasive plants, we herbicide treat them or treat them in some other way, and they go away. The native plants come back, the lake is healthy, yay. But this is not an ideal world, and what we see a lot of times is that even when herbicide treatments are effective, there's usually only a temporary decrease in AIS abundance, and more importantly, native plant community recovery tends to be pretty low. And what this can lead to is reinvasion. So where there's this gap in native plant communities, the invasive species oftentimes just come back, and this creates a kind of a cycle or a loop that repeats itself. So our project is hoping to really address this question of if active revegetation or actively establishing these native plant communities can help us escape this kind of herbicide treadmill. And so previously, MAISRC-funded research has found this relationship between native plant diversity and invader abundance. So in the graph here, you can see on the x-axis, we have abundance-weighted native plant diversity. On the y-axis, we have invader abundance. And for two prominent invasive species, Eurasian water milfoil and curly-leaf pondweed, you can see that as native plant diversity is higher, there's an association with lower invader abundance. And this is just like an association between these two things. It's not causal, but it's exciting because it brings the question of if we were to actually go and increase native plant diversity, would we potentially also see this decrease in invader abundance? And this kind of revegetation approach after invasive species management is a lot more common in terrestrial systems, where after invasive plant management, a lot of times we go in and reseed or repropagate in some way using methods like cover cropping. But this just hasn't been tried out as much in aquatic systems and lakes, which brings us to our project. And since there's some larger gaps in understanding, we're hoping to take this multi-pronged kind of an approach to addressing this question. So the first thing we want to do is we're going to broadly try to identify attributes of native plant communities that can confer this biotic resistance to invaders or reduce invasive plant abundance. And then we want to narrow in and find specific native species that we can kind of focus on and develop propagation protocols for them to increase our capacity for actually putting them out in lakes. And once this is done, we want to see how well this works in the field. So we're going to partner with lake managers and kind of learn while doing through in-lake revegetation experiments. So what does that look like in practice? For our first question of if native vegetation confers invader resistance, we're hoping to analyze large data sets like MAISRC's PI charter, which this is a screenshot from. And this data set, for instance, has information about plant communities in lakes generally that includes both invasive and native plants. And so by analyzing this kind of data, we can start to tease out the relationships between different plants in these plant communities and see if there are specific attributes of the native plants that are more associated with decreases in invasive species. And we're hoping to go beyond just species richness in this kind of analysis, which is just like how much of each plant is, where each plant is occurring and which plants, and we want to kind of look at how much. So looking at species abundance on top of that. We also want to look at environmental data to kind of see if these relationships hold true across different kinds of lakes, and also if there are any mediating factors for lake communities, such as water clarity or sedimentation. And lastly, we're hoping to kind of hone in on specific traits as well for these native plants. And so an example of something we might find is that things like the local abundance of native plants and specific functional characteristics are contributing to biotic resistance and leading to decreases in invader abundance. And so once we've broadly kind of established this proof of concept and this general idea, we want to focus in on specific species. We're hoping to identify 3 to 5 focal species for our trials. In addition to this data analysis component, we want to convene a working group of practitioners who are already engaging in this kind of work and have expertise in aquatic plants to inform our species selection. And then from there, we will conduct experimental propagation trials, like in a greenhouse setting, to increase our capacity for actually, doing this. A lot of plant nurseries don't currently propagate aquatic plants, so even if this seems like a good idea, we don't currently have the capacity to actually go out and implement these either right now. And ideally, this will lead to us partnering with different groups through research management partnerships and increasing our adaptive management of lakes. Here are the logos of a few people who wrote letters of support for us, who we're hoping to collaborate with throughout this project. And they're already doing a lot of awesome work with revegetation in lakes. And so we're hoping to kind of synthesize a lot of the information that's already out there and a lot of the hard work that's already been put in, and see if we can distinguish patterns that already exist. And then after coalescing that data and putting it together with our previous steps, we want to develop a kind of standardized set of installation or monitoring methods that could really help us understand the efficacy of this method and its potential to be used statewide. So the long-term strategy for this project is to provide this framework and best set of practices for enabling aquatic revegetation across the state. We're hoping to provide practical recommendations for people actually looking to do this. And we want to just increase our management toolbox generally for invasive aquatic plants. And so ideally, if we were able to combine this kind of revegetation approach with a lot of the management interventions that we already engage in, such as herbicide treatment, that might move us a little bit closer to our goal of having AS-free, healthy native plant community lakes across the state. Thank you.
Thank you so much, Abha, for that research. I'd like to ask a few questions. We have time for more, so if you do have a question for Abha, please put your question in the Q&A button at the bottom of your screen. The first question is, what attributes and traits of native species do you expect to find will be important in reducing invasive plant abundance?
Yeah, so there's already a lot of good information out there, and people have been working on this, and it seems like abundance specifically of native plants might be a really interesting attribute that we could see related to decreases in invader abundance. And I think that'd be really interesting because, as I kind of mentioned, a lot of previous studies have focused just on species richness and looking at the number of plants in there and not really how much of each plant is in each lake. So that's something that we're hoping to consider in tandem with our other variables that could lead to exciting results. And it'll be really cool, I think, to look at trait level data and see if we could find specific plant traits like carpet growth forms or specific ways in which plants propagate that might be also very useful.
Another question here says, do you already have some good ideas about which species or groups of species might be good AIS competitors?
I do not already have a bunch of good ideas, although that would be awesome. I think we were thinking about potentially, as I was mentioning, like the growth forms of different species. So like Cara and carpet forming growth forms might be really cool to try out and might be like kind of serve that similar cover cropping concept that there is in terrestrial systems, where maybe if you have some animals that kind of quickly take over, that could help fill in this gap left by invaders and prevent further abundance of invaders, but to be determined overall.
And this question says, how would you or someone plant the native plants to restore a lake after large-scale AIS treatment?
Yeah, it's a great question. So we're kind of hoping to potentially address some of this in our propagation trials because aquatic plants reproduce in a variety of ways, right? So they could, through seed, it could be clonally, through rhizomes, through turions. So the kind of growth form that our focal species take and the reproductive structures they have might inform that. Ideally, we would go and plant these in the lakes after we do it in different sedimentation types and environmental variables in our greenhouse setting. So hopefully that'll help answer some of the questions of how, in addition to what